So thank you to the organizers for inviting me to give you this update on the CoA-Z clinical trial. So as you've heard from my colleagues, Dr. Hayflick and Su-Young Jeong, PCAN is a disorder of vitamin B5 metabolism. It causes a severe neurodegenerative disease, particularly in younger people who are affected. The disorder is a result of a derangement of a mutation that causes a block in the pantothenate CoA pathway in this first step. And what we proposed to do based on the work in the lab by Sue and Susan was to investigate 4-phosphopantothene as a study product, a downstream product of intermediary metabolism. This is not a novel idea and concept. It's been tried in many inborn errors of metabolism. And so with the rationale that was detailed in their presentation, I'm not going to give more details on that. The full details are also in this paper that is referenced here. So before we launched into a trial in a large number of people, what we wanted to do was to assess whether the doses that we were proposing to use in humans would be effective in changing our biomarker of interest, and to also take a look at the preliminary safety and tolerability of those doses. The doses were derived from the mouse studies and converted into human doses using FDA and other algorithms for mouse to human conversion. Our biomarker of interest, putative biomarker of interest, is this bifunctional enzyme, CoA-Z mRNA expression of this bifunctional enzyme of this step of the pathway. So this is what our first in-human study looked like. This was a proof of concept study, as I said, to look at preliminary safety and tolerability. We wanted to look at the pharmacodynamic profile of our putative biomarker. This study involved participants, 3 atypical and 3 classic PCAN participants came to Portland for about 8 days. They had had a biomarker drawn sometime in the -5 to -1 day window. They had another biomarkers tube drawn at day 0. They also had safety labs done. And then biomarker tubes were drawn on day 1, 2, 3, 8, 14, and then on at day 28 after they had been off the compound for 14 days. We also did safety labs at various points during this time. We were seeing participants daily in our research center for the first week, and then they went home for the second week while taking the product, came off the product, and had follow-up labs done later. So a 14-day treatment period and 14 days of washout. This is the results from that first in human study. I'm really focusing on the CoA-Z mRNA data here. I will just say that the compound had a very favorable safety and tolerability profile, and there were no adverse events or side effects reported, no lab derangements of significance. The compound looked very safe, and our formulation that we had developed was enthusiastically ingested by our participants with good feedback all the way from our youngest pediatric participants to our adult participants. They liked the strawberry flavor of our compound and found it easy to take. So the results we saw, so the pre-dosing CoA-Z levels were within each participant. What I'm showing you here is their pre-dose and post-dose for each of our 3 atypicals. Pre-dose, post-dose in orange, pre-dose, post-dose in orange here for our classic patients, and then the combined data for atypical and classic patients. And as you can see, we did see a significant rise in CoA-Z mRNA expression And that was very encouraging to us to give us some confidence to go forward with the phase 2 study, that the doses that we were using were reasonable. So in the phase 2 study, this just gives you a little bit of the sort of age range. Our atypicals were 27 to 35 and classics were just 4 to 6 years old. So in our phase 2 study, we then embarked on that. This is a study that is funded by the National Institutes of Health with ancillary funding from a large group of folks that I will acknowledge at the end. It was done in collaboration with Oregon State University, who helped us with the formulation, shelf life study, preparing the product, and Washington State University, who, as Allison has mentioned, is helping us later in the trial with latent growth analysis of our natural history data in collaboration, in concert with these data. So perhaps the thing that I want to talk most about is that we proposed something that really had not been done before, which was a completely novel approach to clinical trial execution. This is a study that is conducted entirely remotely in the participants' own home, own community. They do not travel to a study center. This greatly reduces the risks and burdens of research, but it requires a lot of our participants. And we, wanted to see if this was going to be feasible to run a clinical trial in this way. We had reasons for wanting to do this. We have observed in other studies that when participants travel long distances or really have any stressor, it can sometimes exacerbate their disease and thus confound the clinical trial outcomes. So we had very novel approaches. We developed videotapes, online videotapes that were accessed by private links for pre-enrollment, pre-consent process. Then we did the consent process online. We developed a contract with a national laboratory and then individual arrangements for other participants to do local blood draws. And then every part, the compound is shipped to participants. They ship their empties back to us. It requires a huge amount from our team and from the participants themselves. It reduces the cost of clinical trial conduct, and it would not be appropriate for many drug trials, but because this was perceived by the regulatory agencies to be a low-risk compound, it's essentially a vitamin metabolite, we did eventually get endorsement to run the trial this way. And as a side benefit, it turned out to be completely pandemic-proof. Ours is one of the only clinical trials in the country that really continued without any interruption at all through the pandemic. It really does take more than a village. It takes an entire city to make this happen. We have a small but very efficient team. Our colleagues at the Food Innovation Center from Washington State University have helped us with the formulation work. Of course, our team at the university, our shipping days are these massive events where we ship out dozens and dozens and dozens of bottles to many participants all around the US and Canada. And then, of course, we have our participants at the far end of it who have enthusiastically endorsed this approach. So the study design, it was a classic placebo-controlled double-blind design with 6 months of a double-blind phase, followed by a planned 18 months of open-label phase. Because there was when we designed this, there was another clinical trial going on, and because this is an ultra-rare disease, although we did think people would embrace this study enthusiastically, we allowed ourselves 2 years to recruit participants, 51 participants, so that we would have 12 in each dose group. We were looking at 3 doses either side of our sort of target dose that we had calculated. This is a safety and tolerability trial first and foremost, and then as a secondary outcome, we are looking at pharmacodynamic responses and the profile of the biomarker over time. And then also as a secondary outcome, we're looking at our natural history data in concert with this to detect a disease-modifying effect pre and post-treatment. Now, we anticipated 24 months recruitment period, but this is what it looked like. This is what our timeline looked like. We wanted to get 48 completers, each of whom would have 24 months of study product. But our expected versus actual enrollment, this is either a dream curve or nightmare, depending on your perspective. Usually this sort of putters along. It starts out strong and then tails off over time. Our community was really ready for this. The REO24 study, from our point of view, it closed fortuitously. The timing of that was fortuitous because it enhanced our enrollment, and we enrolled our entire 51 participants in a matter of 2.5 months. So we kind of paused for breath at that point and actually revised the study design. It is still 6 months of double blind, but now we've extended the open label. We've also expanded out to 30 months. So each participant will have 36 months of treatment with the study product. And we are aiming for 60 completers with 15 in each group. So I'm going to talk briefly about the current status of the trial. So the enrollment status, we are now close to meeting our target 64 participants for 60 completers. We have actually 57 on study product and fully enrolled. We've got others that are consented and in the pre-screening process, but we basically have our entire cohort identified at this point. I'm going to just talk about the baseline characteristics briefly. So this just shows you our distribution. We have a relatively even split of less than 18, greater than 18. I realize what I don't have on here is I think I didn't put it in, the classic versus atypical. There is a slight shift towards the classic patients over atypicals. But one thing that is really striking, and which I would be interested to hear people's input on, because we have not gotten much traction on this, but it's something that we're going to be looking at in detail and may actually be a limitation of our trial, is that we have a quite striking male to female bias. So 65%, 35%, this has been the case pretty much throughout the trial. And while we have a fair number of female participants sort of teed up in this for this last push, we are still going to have a gender disparity in our enrollment, which is really interesting to us. We don't have a great answer for that. Just to preempt the question. So participants, I would say more than 60% at this point are through the double-blind phase and are rolling over into open label right about now. The compound has had a very favorable safety and tolerability profile. The compliance with this very demanding trial in some ways for our participants has been superb, really excellent. Everything that could have happened has happened, and yet somehow everybody manages to get their bloods drawn more or less on time, a few lost samples here and there, a few missed samples, but really the overall compliance with everything has been quite excellent. We are now planning, because we have so quickly reached our target cohort, we are planning a direct to open label cohort next so that we can continue to enroll people as they are either newly diagnosed or come to our attention. I would just mention that we have been able to enroll people from the Amish community that Alison mentioned. This took a remarkable effort because they do not have phones, typically, just one pole phone in the community. They're not online. We have to come up with creative ways to be able to work with the Amish community, but through some dedicated providers and a lot of effort on the part of our team, creativity, we've been able to enroll people in the Amish community, and that's been quite wonderful. We've got people from all walks of life who would never be able to participate in a classic design clinical trial. There is a plan to expand the trial to other countries, but these plans are quite mature in the Netherlands, with Odi Sibone leading those efforts. And in the United Kingdom, where Manju Kurian and her colleague Rob Spall are leading those efforts. They are going to be focusing primarily on younger participants, at least to start out. In the UK, the Netherlands design is a little bit different, but the UK design will essentially follow this same design. So with that, I will acknowledge our funding supports, the Spoonbill and Lepelar Foundation. ZZF was contributor to Lepelar. These are largely patient and family raised funds, an extraordinary, inspiring effort from our patient and family community to be able to get us to the point of launching a clinical trial, by which time we were able to secure a large grant from NIH, from the National Institutes of Child Health. We've had additional support from NBIA Disorders Association. Thank you all. And these folks supported our first in human study. Our team, and strangely, Odi managed to be a part of it. This was us on Rare Disease Day. You'll probably see this picture a couple of times during the day. So with that, I will pause.
Thank you very much, Penny. I'm opening the floor to questions, and I'll start with the list that I have here. Now, first question, have you performed any pharmacokinetic analysis of the bioavailability and plasma half-life of the treatment?
So we have not at this point, and that was largely because of limitations in what we could measure at the time. We do have some samples saved. Whether they are going to be adequate for us to be able to do this retrospectively, I think is a question. It is something that we have discussed as a team now that we have a much more robust way of being able to measure 4-phosphopantothene. That took us much longer to get fully validated and underway than we expected. We are talking about how we might best do that. It is not as easy to, this is one of the limitations of a remote design, it's not as easy to fold in some types of samples because we are collecting them remotely. We are limited by what processing can be done on, at the point of collection, unless they can be in a stable tube that can be shipped to us. And so we're looking at how best to do that. It may be another study that is done at OHSU with participants coming to us to do careful PK studies at that point. So it's a little backwards the way people usually do it, but we had an opportunity to move forward with the phase 2 and wanted to go ahead and do that.
The next question is, how does the CoA-Z mRNA pre and post CoA-Z treatment compare in age-matched controls?
Ooh, I might need to fold in Susan and Sue for this, because we have collected quite a lot of samples from healthy controls. This was not a study where we had, so we have not treated healthy controls with CoA-Z, with the compound. But we do have a number of samples from a number of different age-matched controls of CoA-Z mRNA expression from healthy controls to compare to. We considered whether to do a healthy control study and talk to the FDA about that, a treatment study with CoA-Z. And there were some compelling reasons why we might not want to do that. But because of the down regulation that we believe happens with CoA-Z mRNA expression, that might be a challenging study to do in any case. That said, I might let Susan and Sue, if there's a way to do that technologically, kind of address the issue of healthy controls versus the PCAN patients at baseline pre-treatment. It is, I think, included in the paper.
Yeah,
Susan,
so
do
I
you want
can,
to comment?
sure. I actually answered it in the Q&A because Manju had asked in the chat, and then there was also a question in the Q&A. So I've answered it there, but we have, so Sue's paper shows the healthy control versus classic or atypical PCAN. And what we saw in the first in human results, though there's some problems with comparing one experiment to the next, but we saw a recovery, a maximal recovery in the range of what we're seeing for healthy controls. We did see a fair amount of variability by age. So if you binned by decades, age decades, you definitely need age-matched controls. That was very clear to us. But I don't think we've published that full data set, but that will be included in a forthcoming publication.
Yes, one last question here. What is the stability of the biomarkers being measured since they are being shipped?
So we are using a clinical tube, and that does stabilize the mRNA for 72 hours. So we have to get the sample back to us within 72 hours and into the freezer. It can be shipped ambient, which has been helpful to us. And we've had really, we've had a couple of times where we have lost samples because of shipment accidents or participants putting the wrong label on, not putting the overnight shipment label on, those kinds of things. But overall, I think we've actually done remarkably well in being able to get the samples to us within that 72-hour window. It took, I will say that trying to get the tubes drawn properly was definitely a learning curve for our laboratory partner. And because we're dealing with the laboratories all over the country, we had to develop some very specific educational materials for the labs. And again, we made the patient family community our advocates. They would go and put this right in front of the person drawing the tube to say, you need to make sure it goes to this line. Because early on, we found that we had some samples that were not adequate.